This is a video protocol for electroporation-mediated RNA interference method in Odonata.
Dragonflies and damselflies, members of the order Odonata, are among the most ancestral insects. Adult Odonata show a remarkable diversity in body colors and patterns, and many ecological and behavioral studies have been conducted. However, molecular genetic studies on Odonata have been lacking because gene functional analysis is very difficult. For example, the conventional RNA method is not effective in Odonata species. Recently, we established an RNA method in combination with in vivo electroporation. Here we provide a video protocol for the electroporation-mediated RNAi in both dragonflies and damselflies.
As a representative damselfly species, we use the blue-tailed damselfly, Ischnura senegalensis. This is one of the most common damselfly species and often found in sunny ponds in Japan. First, we show the RNA protocol targeting the abdomen of a damselfly, Ischnura senegalensis. After ice-cold anesthesia, attach two pins on both sides of the prothorax and fix the larva to a fixed stand.